So we already talked about these introductory topics about enzyme activity, and now I finish also discussing some regulation such as cofactors and enzyme inhibitors. Now the next step is we're going to talk about allosteric regulation. And so we will move to study how the regulation of an enzyme activity helps to control the entire metabolism. And to that we will talk about first allosteric enzymes. And to do that I'm going to show you two videos that they summarize very well what is allosteric enzymes. Then I will move to talk about allosteric activation and inhibition. Finally, I will talk about cooperative activation, and finally, I will finish talking about feedback inhibition. So let's start. To start this topic of the regulation of the metabolism, in other words, all the chemical reactions taking place in a cell, in this diagram, I want that you see that chemical reactions have two types of regulation. One is through gene expression, and the other one is through enzyme activity. And in this case, the example is the regulation of an amino acid called tryptophan. And if you see here, there is a negative feedback. There is a regulation. When you have too much tryptophan, the enzymes are not going to have as much of this precursor, so they are going to start reducing the amount of enzyme activity. And in a similar way, there is a genetic regulation. In this case, that when you have too much tryptophan, there will be a signal that doesn't allow the expression of these genes that are going to produce these enzymes. So in the first case, we talk about the regulation of an enzyme activity. And in the following slide, I will show you how this tryptophan can inhibit all this enzyme activity. The following slides, and I will show you a video. And I also show you that genes are also important to regular enzymes. And why we have this such a tight regulation? Because if we have a chemical chaos, the cell will die. Yeah, so they should be tightly regulated. Second thing that I told you, the one that you remember, is that a cell does this switching on and off of genes that encode a specific enzyme. And this is one way. This is the second way that you can regulate enzyme activity. And the third way is by regulating the activity of the enzymes. And I will show you how this feedback negative can be a way to inhibit and regulate the enzyme activity. Now let's talk about what are allosteric enzymes.
Our cells are amazing molecular machines. Every day, they convert food into energy, such as ATP, that drives the millions of biochemical processes necessary for keeping us alive. The pathways used to convert substrates, such as glucose, into products are collectively referred to as metabolic pathways. The drivers of these metabolic pathways are enzymes. Enzymes are usually proteins that work to assist chemical reactions by building or breaking down molecules. However, enzymes don't drive reactions at a constant rate. The rate of reaction can speed up, slow down, or stop completely according to the cell's needs. This process is analogous to an assembly line where raw materials are converted into products. The assembly line must be monitored to ensure that the supply of products does not exceed demand. If products are being created at a rate that is faster than they can be used, both resources and products are being wasted. The assembly line can produce at a slower rate or stop completely to prevent waste. In cells, this process is called feedback inhibition. Feedback inhibition is achieved through a process called allosteric regulation and plays an important role in many metabolic pathways. Enzymes have a unique area called the active site where substrates can bind. Enzymes that can be regulated have an additional area called the allosteric site. Downstream products can bind to this regulatory site, causing the enzyme to change shape and prevent it from binding with its substrates. This causes the entire pathway to halt and new products are no longer formed. Conversely, when the amount of downstream product gets too low, the enzymes interact with their usual substrates and begin forming products again. Feedback inhibition plays a critical role in preventing cells from wasting resources and energy. Mutated enzymes that do not respond to allosteric regulation have been linked to disease states such as cancer. Many processes in our bodies rely on feedback inhibition to keep everything running smoothly and efficiently, maintaining homeostasis.
Let's talk about difference between simple enzymes and allosteric enzymes. Simple enzymes only have one active site, whereas allosteric enzymes have an active site and a regulatory site. Simple enzymes are substrate specific, whereas the allosteric enzymes are specific for the substrate and a modulator. In case of simple enzymes, the rate of reaction decreases in presence of competitive inhibitor, while in case of allosteric enzymes, the modulator may increase or decrease the rate of reaction. Simple enzymes shows hyperbolic curve, whereas the allosteric enzymes shows typical sigmoidal curve. Simple enzymes are also termed as non-regulatory enzymes, while allosteric enzymes are termed as regulatory enzymes.
Now let's talk about allosteric activation and inhibition. And to do that, I want to show you this diagram of your textbook. And as you can see here, you have 1, 2, 3, and 4 polypeptide subunits. So instead of having one, you have multiple. And you not only have multiple subunits, you can have multiple active sites. So here you have 4 active sites. And you have also 4 regions to regulate the activity of an enzyme. So in this case, for example, you can activate. So you can have an activator that attach to this region, and in this way this enzyme will stabilize and will have these active sites open to be able to interact with the substrate. So now let's go and recap all the things that I just told you. And the first thing that I told you is that most allosterically regulated enzymes are made of polypeptide subunits. So here you have 4 subunits, and you not only have 4 subunits, you have 4 active sites and also 4 regulatory sites. And they are constantly oscillating between an active and inactive form. And I'm going to show you an example of an active form. So when you have an active form, you can have an activator that stabilizes the active form of an enzyme. So for example, this activator is going to attach here in the regulatory site, and now this enzyme is stabilized in the active form. Now in the following slide, I'm going to show you the other example of the allosteric inhibition. So here we have the previous example of the allosteric activators. Now I'm going to show you that they are oscillating constantly between active and inactive. And here, instead of having an activator, you're going to have an inhibitor. And this inhibitor is going to attach in one of these regulatory sites. And if you see, these active sites are already inactive. When the inhibitor attached to one of these regulatory sites, it will stabilize the inactive form. Let's recap again all the things that I just told you. So here, as I said, there is a non-functional active site. The substrate cannot go and attach here. And when you have an inhibitor, the inhibitor is going to attach into a regulatory site, and then this will stabilize the inactive form. Now I'm going to move to talk about another type of allosteric regulation, that is the cooperativity. And this is another type of allosteric activation. In this case, you go from an inactive form to an stabilized active form, but instead of using a regulatory site, you use is a substrate. So this substrate change not only one active site, but change all the other active sites. So now much more substrate can have a high affinity and you can speed up the chemical reaction in this way. So you can have a change reaction that the substrate is allowing to have a lot of activation. This is why it's cooperative behavior. So let's recap. Cooperativity is a form of allosteric regulation that can amplify the enzyme activity. The second thing that happens is that one substrate can prime an enzyme to act on additional substrates. So here, the additional substrates here, here, and here to act. And this, how is that? Because the binding of a substrate to an active site affect the catalysis in another active site. So as this one bind, I show you that this one, and this one also change. All these four regions change because only this one changed. And this was the cooperative allosteric regulation. Now I would like to show you a video of another type of regulation, that is the feedback inhibition. So please watch it.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
So now to summarize what we watched in the video, I have this schematic. Here we only have two enzymes, 1 and 2. And in this case, let's suppose that we have a substrate. And suppose that is a polymer, glycogen. Let's suppose that is glycogen. So you have a lot of sugar. You have many sugars, and now your body say we need to get monomers, and so you need to produce glucose because you need energy. So what is going to do the enzyme? The enzyme is going to create an intermediate substrate, and then you have another enzyme, and then you get your glucose. However, there is a point that you have so much glucose that your body says, we don't want to waste, we don't need more glucose, we don't need to waste all the glycogen. This is taking too much energy and we're okay. So what is going to happen is that this end product is going to attach to the enzyme and is going to modify, for example, in this case, the active site, and is going to inhibit that this process continue. So the feedback inhibition, the end product of this metabolic reaction shut down the pathway. Here, for example, the glucose shut down in this case, this green molecule that for example, it could be a polymer like glycogen, that it needs to be broken into monomers. Now this is not happening anymore because of the feedback inhibition. And as I said, the second thing that I want that you remember is that the feedback inhibition prevents a cell from wasting chemical resources. And how it does it? By synthesizing more product that is needed. And this is the last idea that I want to tell you today. And if you have any question, just go to your textbook and start trying to solve all the exercise of the dynamic modules, all the things that we have to do. And if you have any question, we will solve that in class. Have a good day. Bye.